Good afternoon, everybody. Hi, I'm Sangeeta. Welcome to the session Nanomedicine for All. We have a great session planned for you today. I'm going to start by just giving a little bit of an overview about what we mean by nanomedicine for those of you who aren't used to thinking about nanotechnology in medical terms. And then we're going to have three fantastic speakers, and we're going to close out with a panel. Okay, so for this crowd, I think you're well familiar with the idea of nanotechnology emerging from a combination of top-down fabrication and bottom-up synthesis. And collectively now, we have nanoscale materials that have these fascinating properties that emerge: optical properties, electronic properties, magnetic properties, and this is all of your world. What's been really exciting for those of us like me, who are trained as a physician as well as an engineer, is that when you make materials this small, you can open up new areas of biology and medicine. These materials are now right-sized to talk to biology, and there are emerging nanoscale biomedical properties as well as these sort of physical properties. So what does that look like? First of all, on the left here, you see that there are a number of materials that have emerged that have made it all the way into clinical trials. So these are materials that were synthesized on the bench, characterized, looked at for their safety. They had these emergent properties, and they're now in humans. Many of you actually got one of these nanoparticles in the mRNA vaccine. Those are lipid nanoparticles. So those are materials that have been in millions of people. And what's so amazing about them, if you look at this sort of molecular ruler, is that the nanoscale is actually the length scale at which biology happens. DNA is nanometer scale, a virus is 100 nanometers, a cell is about 10 microns. A single receptor is something that you can interrogate with one of these nanomaterials. And as a community studying nanomedicine, which is nanotechnology applied to medicine, for about 20 years, researchers around the world have been discovering new properties. And on the right here, you see some of the properties that we've discovered about biological nanoscale systems. For example, materials of different size at the nanoscale, when injected in the human body, will traffic to different organs. A 100 nanometer particle will go to the liver. A 3 nanometer particle will go to the bladder. The shape of a material can affect whether a cell internalizes it or not, if it's long and floppy or if it's small and hard. You can make materials that actually are activated by the human body, by enzymes or low pH or oxidative states, so they're bioresponsive. And when you put materials in the body, they actually get coated with proteins and biomolecules, and that those coatings, those interactions, can then direct where the nanomaterials go. So the combination of nanotechnology and medicine is giving rise to this whole set of new insights and new capabilities that we are collectively really excited about, and I hope you can learn a little bit more about today. I just want to give you sort of one last snippet, which is that this is an incredibly exciting time for those of us in nanomedicine, and that's because there's a convergence of decades of science coming from two pretty disparate fields. So on the top here, you see the convergence really of fabrication technologies and the speed at which those have accelerated. Of course, this has really been driven by our interest in computation collectively, and many of you work in this space. But what you may or may not know is there are areas in biology and medicine and chemistry that have been quietly accelerating on their own. And here I give just one example, which is the example of RNA. RNA being advanced as a therapeutic, RNA interference, mRNA, packaging of RNA into nanomaterials. And those two things really came together over COVID to scale up materials and, as I said, to show that they were safe in millions of people. Now these same materials are being used to enable CRISPR, or genome editing. So if you look at the convergence of these timescales in this moment of time, it's incredibly powerful to think about how we get to leverage all of this engineering and all of this biomedical work and think about ways to combine them. I run a center here at MIT, the Marble Center for Cancer Nanomedicine. Kurt is actually here today. And we have a fantastic group of investigators. We're localized in the Koch Institute. We have collectively over 200 community members thinking about some of these problems. So please come visit us. We're super productive. We publish a lot about this. We patent a lot. We've had a lot of startups, which some of you may be exposed to. And some of the ideas that we are really excited about right now are listed here. The idea that you can intercept cancer before it's lethal. The idea that you might be able to vaccinate in a personalized way against cancer. The idea that you can precisely treat cancer by knowing more about its genetic code. And finally, really the theme of today is how we can take all of these discoveries and make them accessible to patients all around the world, which is actually not the case today. As you know, the lipid nanoparticles that I described require a cold chain. They require being frozen at minus 80 degrees. So we really want to think about how we can enable these medicines spreading all around the world. So as I said, come visit. Today we have some community members here that you're going to hear from. We're going to start off with Mujibu Wendy, and then Angie Belcher, and then Bob Langer. And then we're going to have a panel and take questions from all of you. So with that, I'm going to briefly introduce Muji. He is the Lester Wolf Professor of Chemistry. He trained at Harvard, the University of Chicago, and Bell Labs before arriving at MIT in 1990 to join the Department of Chemistry. His work on the basic concepts in material science, synthesis of high-quality nanoparticles and quantum dots led to the 2023 Nobel Prize in Chemistry. His lab's research ranges from the very fundamental applications in electro-optics to applications in biology. And he's going to tell us in just 10 short minutes about what he's thinking about now. So welcome, Muji.
Well, thank you, Sangeeta, and I'm really delighted to be here. I'm going to tell you about this journey, start in the mid-1990s, when the field discovered that we could make really bright quantum dots, multiple colors with an absorption where that was continuous into the visible, and especially at 465 nanometers, which is where the blue gallium nitride diodes had just been invented. And so that meant that we can think about multiplexing using one of these newfangled blue diodes. And if we could target different parts of cells or do in vivo biology, then we could image with one light source, but if only we could get the quantum dots to be compatible with biology. So we were able to do that in the late 1990s. And then that's when I first met Bob Langer, because I went to his office and we were about to start Quantum Dot Corporation. I had no idea what I was doing. And so I went to Bob and asked him for advice on what to do with starting this company. So I started this company with Paul Rosados and Shimon Weiss at Berkeley and a couple business. So this company's created the first product of quantum dots coded so that they would be able to target cells. This was an example from a paper from that period of time looking at a cell, comparing Alexa 488, which is a dye labeling the skeleton of the cell, and then the nucleus being labeled with a quantum dot showing the difference in the photobleaching. That is actually a hard application of quantum dots, but we pursued it. The easy application is all over the place today. If you have any Samsung screens somewhere here, they're full of quantum dots to get the beautiful green and red colors at low efficiency. So as a side note, if you create something that's more energy efficient, humans find a way to spend as much energy. So we just make it bigger and brighter, and it uses as much energy. So you have these beautiful, big, bright screens, thanks to the quantum dots. But anyway, we had to learn how to handle nanomaterials in the biological environment. We had to learn how to make them non-sticky so that we could inject them in this mouse model here. The reds are the quantum dots, the green is GFP, so that they would be able to get out of the vasculature into the tissue so we could access things that we wanted to target. So we had to learn how to coat these things with PEG or zwitterionic molecules. We realized that size was incredibly important. We did some biodistributions and realized that if you wanted to have something that could go in there, target, and clear, it had to be probably less than about 6 nanometers in hydrodynamic diameter. Otherwise, it'd go to the liver or just stick around for a long time, and you'd get this background. And so we worked very hard to make them as small as possible. And also we worked hard to understand the effect of shape. This was work done with Rakesh Jain at MGH. So we looked at different sizes. This is in a tumor microenvironment, going from 12 nanometers to 125 nanometers in size. And you look at the penetration depth as the distance from the vessel. Up here, you see that the big ones, 60 nanometers, they just don't go into the tissue at all. They just stay in the blood vessel. So in terms of delivery, that's terrible. The 12 nanometer one is able to penetrate out of the blood vessels into the tumor. Then if you look at the effect of shape also, so these are two particles of different shapes. The morphology is the same volume. But if you look at the rod one, it's pretty long, but it's pretty narrow, you find again that the tumor transport for the rod, the red bar here, is much better than the sphere, even though they have the same volume. So it's the small dimension that really matters in terms of being able to go through the vasculature and into the tissue and being able to diffuse. So we learned how to target things in vivo in ways. So these are rare hematopoietic precursor cells that, generally before, in the old days, you'd have to take them out, label them, and put them back in if you wanted to study them in vivo. But here we were able to show that with a multicolor imaging, we could take quantum dots of two different colors, which then go to receptors that are overexpressed on these particular cells jointly. They're not, they don't label blue, Herx blue. So what you're really looking for is yellow then in your microscope. And then you can find these very rare cells in the bone marrow of this mouse model, and then you can look at different ones of these and see where they reside, and eventually one would like to understand about the microenvironment of these particular cells. And so doing this in vivo is super important because then you're not affecting them, you're not differentiating them, you're looking at them in their environment without disturbing them. This is kind of a hero experiment. And so we thought, what are dots good for that dye molecules aren't good for? One of the things it could be good for is to go into the infrared, where tissue is less scattering. And so we worked again with Rakesh. We created these composite particles that are about 100 nanometers. Why do we do that? Is because you can actually see the individual particles. So if you want to track the individual 100 nanometer particles that are fluorescent, you can actually see them in real time. So we used regular dots, the 4 or 5 nanometer dots, to label the arteries and the veins and the tumor itself. This is actually separated in time because the arteries come first. Then you wait, and the veins come out, as the particles clear. And then you wait more, and then everything clears except what's in the tumor because it gets stuck in the tumor. And so you can label then the different time snapshots, different colors to label different things. Then we can go in with the composite particles. Now we know where to look. And we can look at the diseased tissue versus the normal tissue. And then we can see the difference in flow. And you see in the normal tissue, very fast flow. These are the little white dots are the individual 100 nanometer particles using, basically night vision imaging. And in the diseased tissue, you see the flow is really not great, right? And so this is a visual way of seeing why it's hard to treat cancer. You can't deliver it. You can't deliver therapeutics. Rakesh was interested in figuring out how to first use a therapeutic to change the flow from something that was really pendular, not going anywhere, by snipping out the growth of small blood vessels, for instance, to make it more like the normal flow, so you can actually deliver things, you can make it less acidic, get oxygen in there and all these things. And so I really enjoyed this, but then, I was always interested in going into humans, and so we had learned a lot from doing all this over the years. We applied this to FDA approvable stuff. This is the story of Lumicell, which was funded by the Marble family, seed funded initially back in the early 2000s, or late 2000s actually. And so we created this nanometer size object with a dye quencher pair, so that attached with a peptide, so that in the presence of a solid tumor, this peptide gets cleaved by enzymes that are overexpressed in the tumor, releasing a dye molecule which is fluorescent. And what we learned from the quantum dot work was all about clearance and biodistribution, because given the specification of the surgery, we knew that we had to have the injection early morning. The surgery had to be a few hours later. You couldn't have the injection the night before, because patients often just come in for outpatient surgery. And so it had to become active within a couple hours. Then it had to clear by the end of the day, within 6-7 hours. And so that defined the size of the molecule, right? And had to clear through the kidneys. And so by defining the size and defining all the little pieces, we were able to create this agent. We combined it with a handheld imaging system that came out of our work on being able to see single quantum dots. We knew how to do things very sensitively. And I'm happy to say that after a long period, this company finally got FDA approval for fluorescence-assisted surgery back in April. So now they're busy scaling it up and hopefully going to a commercial product for helping breast cancer patients very soon. Basically, first quarter of 2025. Another thing that I was interested in was also this issue of gadolinium, which I think doesn't get enough play. What gadolinium is used in MRI imaging. I mean, it works really, really well. The radiologists love it. It's just amazing. But it's got some problem. Gadolinium is not a normal element that you use in the body. And so it's been shown to accumulate in the brain if you have multiple sessions of this agent. It's an environmental problem, like the Boston Harbor, Tokyo Bay, San Francisco Harbor. They're full of gadolinium from the hospitals that are around it, and the patients get injected, they pee it out. And it turns out that iron oxide, if you make iron oxide really, really tiny, and we made this really tiny ligand that's ionic so that we could keep the size to sub 5 nanometers in the hydrodynamic diameter, you can create an iron oxide that looks like gadolinium. It reacts the same way. It's a positive contrast. So you can see the injection, you can see the arteries, and then the bladder showing up. In this case, these are slightly bigger agents. About 23 minutes later, the bladder is full. You can see this beautiful imaging in this mouse. And then if you make it, then you can look at the biodistribution and see that most of it comes out in the urine. There's a little bit in the liver. And if you make it even smaller, you can then try to think about actually targeting with these small iron oxide particles. So now this is even faster. So by 10 minutes, it's already gone into the bladder. This is mimicking the biodistribution of gadolinium very well. This is after 5 minutes, it's already going into the kidneys. And then depending on the size, you can decide either to do imaging of the blood flow, or you can decide to target into tissue. We applied this first with Peter Caravan and Alan Jasanoff at MIT to look at liver fibrosis. And so this is an image of, on the right-hand side here, my right-hand side is a standard chow mouse, and then the left-hand side here is a mouse that's been fed so that it has this fatty liver and all this collagen. And you can see then the binding, so these iron oxide particles are bound with peptides that bind to the collagen, and you can see then the liver show up with these little particles without using any gadolinium. So anyway, that's the end of my talk. Learning how to use the nanoparticles was super important, how they interact with the body, and I'm sure you're going to hear more from the other speakers.
Next, our speaker is Angie Belcher. She is the James Mason Crafts Professor of Biological Engineering at MIT. She received her PhD from UC Santa Barbara in chemistry. Her work on novel nanoscale materials has been recognized by many prizes, including the MacArthur Genius Award and the Lemelson Prize. Her lab seeks to understand and harness nature's own processes in order to design technologically important materials in medicine, which you'll hear about today. But as you all may well know, she also works on energy and the environment. She's a close collaborator, and it's a real thrill to have her here. Welcome.
Thank you so much. It's an honor to be part of this panel. What a great topic and a great panel. I have been working on nanomaterials my entire adult life. I have been fascinated with natural materials like abalone shells, how abalone make materials. These are biomaterials made by an organism. The reason they're so beautiful and so tough is because they have a protein that makes it and controls this type of material. I'm really lucky to work with Sangeeta and part of the Marble Center and Paula Hammond as well, two of my best collaborators. And I'm going to talk about some of the work that we're working on today. So I work at the interface between nanotechnology and biology and in applications. And so we work on energy, healthcare, and the environment. And just like this organism, the abalone used proteins and used DNA to make proteins to control exquisite control over calcium carbonate nano-based materials, what our lab has done is evolve organisms to have proteins to genetically control anything we want on the periodic table. And we've worked on, I think, 135 materials in the last couple of years that now we can genetically control at the nanoscale. So I always say I've never been accused of focusing, and we focus on making these nanomaterials for all these different applications, from energy to the environment to therapeutics, and we've spun out several companies on this. Today I'm going to focus on imaging. We work also, like in Professor Buendi's very beautiful talk, we also work on imaging with nanomaterials, and we also work on imaging without labels, so label-free imaging. And we use hyperspectral imaging and hyperdiffuse imaging in near-IR1 and near-IR2. So we're interested in imaging between about 800 and 1800 nanometers. And hyperspectral imaging has been used for a long time for non-destructive food inspection and remote sensing. But when you shift to the near IR, when you shift to imaging above 1,000 nanometers, you start to be able to see things at much greater depth, as Professor Buendi showed a few minutes ago. And we developed a technology called Dolphin, which we can look up to 8 centimeters inside of a tissue or an animal to look for tiny tumors. And we develop these data cubes. We do 3D reconstruction on it. I wanted to tell us, well, okay. So one of the things that's really interesting, as was referred to before, is when you start to shift to these wavelengths, there's less dyes and probes that you can use. So there's organic dyes that are in the IR. They don't tend to be as stable. There's quantum dots, which we learned a lot about. There's single-walled carbon nanotubes and rare earth nanoparticles. And so we're using an indium gallium arsenide detector to look at tissue and samples at depth. And so in my lab, we've worked on developing many different kinds of nanomaterials to be able to look at these. Some of which are making single-walled carbon nanotubes brighter. Some are using plasmon-enhanced near-IR1 dyes to look for tiny tumors in ovarian cancer, as well as rare earth nanoparticles for doing single-cell tracking. I want to show you an example of something that we went from idea to clinic in 3 years. And this is a collaboration with my postdoc, Jeremy Lee, shown here. This work was all funded through the Marble Center at MIT. We actually applied for NIH funding for this twice and got rejected. But the idea was that we could use short wavelength infrared and orthogonal polarization to see lymph nodes. And so what you see up here is this is human tissue where there's a lymph node and you really can't see it. But if you're imaging it at particular wavelengths and particular polarizations, you can see the difference in contrast for the lymph nodes. And so what Jeremy and Cision Vision did, and we won a Best 2023 invention and also a moonshot from President Biden for this, is we invented a technology where when a patient's undergoing colon cancer, they take a bunch of tissue out. And they take a bunch of tissue out and they probe with their fingers to find lymph nodes. And they have to look for at least 10 different lymph nodes. It's different for different kinds of surgery. What Jeremy did was invented a technology that when that tissue comes out of the patient, it goes over to a technology called Envision, where you can quickly count how many lymph nodes there are. You go, okay, here's 10 lymph nodes. I don't have to go back and get more. And so it actually really speeds up the process of looking for lymph nodes and classifying them for disease. And so It was idea to clinic in 3 years. We're now in 15 hospitals and multiple companies and looking at breast cancer, colon cancer, and prostate cancer. And so this was all based on this near-IR imaging. And so there's all these lymph nodes in the body, and the idea was to use a combination of near-IR and polarization to be able to find the right wavelength to be able to pull out these materials. And this is in a mouse. You can see the lymph node here. You can actually see, this is all without staining. You can see lymph vessels. Here we're looking at adjuvant response during monitoring in real time in mice, and this is before the adjuvant and 24 after. So it's a very non-invasive way of imaging animals or people samples. And then you can really get a lot of new information about lymph nodes inside the body and look at size, symmetry, border clarity, vessel patterns, and from there start to try to understand and predict disease. And we've done that in triple-negative breast cancer, where we have animals that we're monitoring on a day-to-day basis or twice a day, monitoring changes in the lymph nodes. And what we see is that there's this great increase in lymph node size in the animals that end up becoming metastatic. Okay, that's that story. That's all label-free imaging. So I'm driving to work one day, and I'm listening to the news, and I hear about this this asteroid called asteroid Bennu. Have you heard about this asteroid before? So I don't work on asteroids. I would love to work on asteroids. I do not. But what NASA did was they sent a probe 63 million miles to an asteroid that was traveling at 63,000 miles per hour and is about 1670 feet across. They landed on it. They grabbed rocks from it. They did a biopsy of an asteroid moving in space, collected them, took pictures, and sent it back. And you can see the images. I mean, there it is. They have pictures of it landing. And this is an incredible feat. And it cost $1.16 billion, which was well worth it, right? And yet, in 2024, we cannot go this far inside of a woman's abdomen to find a 1 millimeter tumor. You can't tell me that's not possible. If we can biopsy an asteroid, surely we can look this far inside of an abdomen to find a tiny tumor. So I think that we just haven't tried hard enough. We haven't had enough engineers working on it, and so we think it's unacceptable. So here's the team that's working on this. You all know Sangeeta, Paula Hammond, Kripa Varanasi, and our collaborator Becky Stone, who's head of gynecological oncology at Johns Hopkins. We think that you should be able to diagnose ovarian cancer at much early stages, at stages where you can also intercept it. So why ovarian cancer? One woman is diagnosed with stage 3 or stage 4 every 3 minutes worldwide. The 5-year survival rate is 39% for stage 4 and 17% for stage 4. And the survival rate has not improved in 20 years. Okay. The rates haven't improved, and there's a lot that needs to be understood about ovarian cancer, and so I am not accepting that. So there was a large-scale clinical trial out of the UK recently, which said that patients that underwent CA-125 blood test and transvaginal ultrasound, there was no increase in survivability, which means there is no screen for ovarian cancer. And again, as an engineer, you throw up your hands, you said, impossible. There has to be a way of being able to detect it early. Here's another thing about ovarian cancer is that ovarian cancer doesn't come from the ovaries. 85% of ovarian cancer comes from the fallopian tube. It's a fallopian tube origin. It comes from the fallopian tube. It sits on the fallopian tubes for sometimes up to 9 years before it starts to become cancer and starts to metastasize. We have a window of years to be able to detect it and intercept before it becomes stage 2, stage 3, stage 4, when it is treatable. And so what happens is you have a little mutation called a p53 mutation. It starts to come into something called a stick lesion and then develops into ovarian cancer over a year's period of time. Now, we also know that women that have their fallopian tubes taken out, there's also another large-scale clinical trial out of Canada. Women that have their fallopian tubes taken out after childbearing years, 0 cases of ovarian cancer. It's a way of preventing ovarian cancer is removing your fallopian tubes, which says that it's another way of showing that it comes from the fallopian tubes. So we've embraced the goal of developing accessible detection and diagnostics technology for ovarian cancer in the next 5 to 10 years, and that's with my partners, and we won't accept failure on this. This is something we plan on doing. One of the ways we're doing it in my lab is we're taking that technology that we talked about called Dolphin earlier, and some of the work from Envision, and we've developed something called OvaSeq to be able to scan fallopian tubes that come from the OR. So it was during the pandemic, and I was on a Zoom call, and I met Becky Stone, who's a surgeon at Johns Hopkins, and she was saying that when a woman has her fallopian tubes taken out, sometimes opportunistically as a way of preventing ovarian cancer, or if she has a genetic mutation that leads to ovarian cancer, that they take the tubes are like about this big. They take the tubes and they embed them in wax, and they cut them, and they look at 0.1% of them. She goes, if only we had a way of looking at a whole fallopian tube. And I'd never met her before, but I instant messaged her, which is advanced technology for me, and I said, we can design a system to do that. It's actually pretty easy. So we adapted Dolphin to OvaSeq. So now when a patient undergoes, has her fallopian tubes removed from Johns Hopkins, they're put into organ transport media and FedEx to us overnight, where we scan the whole fallopian tube, either using nanoparticle probes for particular markers for precancer lesions, or collecting enough data using hyperspectral imaging to try to look for interesting spectral characteristics that would show us that the pathologist needs to go in and look here. I was really overconfident. I'm a professor, so I don't actually do any of the work. I was overconfident that my students could actually do it, and they did. We also use these IR dyes, which are not very bright or very stable, but when you complex them with nanoparticles of gold, it actually makes them much, much brighter. And this is the team that is working on these, and our idea is to move diagnosis back to where it can be treated much easier and much better. Here's what a fallopian tube looks like. It just came out of a patient. It comes to us. We track it with Asset Panda. We start imaging. And so when I would go to these meetings, radiologists would say fallopian tubes are invisible. You can't see them by contrast-enhanced CT. You can't see them by ultrasound. They're invisible. And I'm like an engineer. I'm like, okay, they're not invisible, right? You have to be able to find them. And so when you find the right wavelength of excitation and the right wavelength of emission, they're not invisible. So we hyperspectral scan them. We find these wavelengths that works, and so now we can follow them. And this is a patient that we scanned. We found a region, and we marked it. We sent it back to Johns Hopkins, where they took it and they went to our mark. They did pathology on it, and they found an abnormal region that was 250 micrometers in size. So you would have never seen it otherwise. So we scanned a 5-centimeter tube and found a precancer lesion that was 250 microns. And so you say, well, that's kind of cool. What do you do there? Well, now Sangeeta is going in and trying to find biomarkers for these. So we're doing the imaging. Sangeeta is looking for biomarkers and ways of amplifying the signal, so you'd be able to tell from urine, blood, or other fluid that you actually had a precancer lesion. Professor Varanasi is going in with technology he's developed to pick up some of these cells so we can culture them live, because there are no cultures live of these precancer lesions. And Paula Hammond is working on developing layer by layer therapeutics, immunology therapeutics that would actually work on trying to treat a very early precancer lesion, or maybe even prevent it. We're also looking at vaccines. Can you develop a vaccine for a precancer lesion so that the patient never develops ovarian cancer? So we also are on a platform. This was developed by Dr. Gross at Johns Hopkins and Pitt, to where we're trying to turn on its head, like the Henrietta Lacks idea of your not having your control of your own samples. So there's an app that the patients get. They know that their fallopian tubes were taken out. They see them going to MIT. They see images of their fallopian tubes. They can interact with the scientist, and they own their own biospecimens. They track their own biospecimens. We say this is bed to bedside and back again, but it is a team effort. It's a team effort of all these scientists and engineers, physicians, and the patients. The patients are an active and important part of this. And so thank you for your attention.
You can see why we're having so much fun in the nanomedicine space. Last but not least, we're so excited to invite Bob Langer here. He is one of 9 institute professors at MIT. Bob trained at Cornell and MIT before joining the faculty in 1978. His work on materials for sustained drug delivery and engineering new tissues has been recognized by so many awards, but I'll just list a couple: the U.S. National Medal of Science, the U.S. National Medal of Technology and Innovation, the Queen Elizabeth Prize for Engineering, and the Breakthrough Prize in Life Sciences. Bob's patents have been licensed to over 400 companies across pharmaceutical, chemical, biotech, and medical device sectors, and he is the most cited engineer in history. A current focus of the lab is the study and development of polymers to deliver DNA and RNA and genetically engineered proteins continuously and at controlled rates for prolonged periods of time. It's a great pleasure to invite Bob up.
Well, thank you. Boy, I really enjoyed the talks. That's just great work that Manji and Angie are doing. So I thought I'd tell you a little bit about how I got started in this area, which was very, I think, kind of unusual. So my background is chemical engineering. And when I got done with my degree in 1974, you know, for those of you that were old enough, that was 50 years ago, there was like this gas shortage. And if you lived in Boston, you actually had to wait in line for 2 hours to get your car tank filled up. But the consequence of that is that if you were a chemical engineer, you got a lot of job offers from oil companies. And pretty much all my classmates, that's what they did. And I thought that's what I'd do too. So I actually got 20 job offers from oil companies, 4 from Exxon alone. And really, I wasn't that good at all, but they just had so many openings. But when I went to some of these, they tell you, if you could just increase the yield of one of these petrochemicals by a tiny percentage, they said that'd be wonderful, it'd be worth billions of dollars. But I remember flying back to Boston that night from Texas, thinking to myself that I really didn't want to spend my life doing that. So I started to think about other ways I could use my chemical engineering background to help people. And I won't go through the whole story, but in the end, what I did is I had this idea of maybe doing something in medicine because nobody would hire me for a lot of other things. So I wrote to a lot of hospitals and medical schools, and none of them would write me back. But finally, one of the people in the lab where I was said to me, he said, Bob, there's a surgeon in Boston at Harvard and Children's Hospital named Judah Folkman. And he said, sometimes he hired unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But Dr. Folkman had this idea that if you could stop blood vessels, you might be able to stop cancer, and that it was actually mediated chemically, which turns out it was a super controversial idea. Since I didn't know anything about biology, I thought it was a great idea. But at any rate, one of the biggest problems, or the biggest problem, was there's really no way to study blood vessels, because wherever you'd go, there were background blood vessels, except for a couple places, like the cornea of certain animals, and the chicoryal and membrane had actually very few. So our idea was we put certain tumors in these tiny little animal eyes, and then we need which would cause blood vessels to grow, but then we needed a tiny delivery system, like a nanoparticle or microparticle, that could deliver large molecules. Dr. Folkman's theory was that they were all, and mine too, was that they were all large molecules that would do this. So at this time, 1974, the idea of having an advanced drug delivery system, was pretty much unheard of. There was only one company in the world, a company called Alza, that was developing these systems, and they had none approved, but later they'd come out with what are called transdermal delivery systems and things like that. Dr. Folkman was on their board. He went out there, he asked them, could they help? But they all told him it was impossible. They basically said that the molecules that we were talking about were too big. It's like walking through a wall. So, but if we were going to solve the blood vessel problem, we had to solve this bioassay problem. So I spent several years working on this, experimenting with different materials, lipids, polymers, other things, different designs, different procedures, and I actually found over 200 different ways to get this to not work. But eventually, we were able to make little microparticles, like shown on the left, or nanoparticles, cut in half on the right. And we published in the journal Nature in 1976 for the first time that you could release any large molecules, also the first study that ever showed you could deliver nucleic acids, which would include DNA and RNA. Now, I would like to tell you that this met with, people were very excited about it. But in fact, it was kind of the opposite. I remember 3 months before this, I got asked, I was giving a talk at a major scientific meeting. It was actually the first talk in my life that I gave at one of these meetings. It was 48 years ago in Michigan. And I practiced this talk for weeks into a tape recorder. And finally, the day came, and I got up and I gave the talk. And I felt I did all right. I didn't forget too much what I was going to say or stammer too much. So I thought when I was done with this talk, all these older engineers and chemists, being nice people, would want to encourage me, this young guy. But when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, well, we don't believe anything you just said. And this, again, they said the molecules were too big, and the solvents we were using, like methylene chloride and ethanol, would destroy whatever we put in. And actually, things went downhill for me from there, if that were possible. I tried to get research grants to support my work, and my first 9 were very soundly rejected. And then, I loved being a postdoc at Children's Hospital, but a lot of my friends said, maybe you shouldn't do that your whole life. And so I did. I started looking for faculty jobs, and since I'm a chemical engineer, I applied to chemical engineering departments. No chemical engineering department in the world offered me a position. I finally got a job in the nutrition department here. It's a long story. I won't go all into that. But anyhow, and then I had some other ideas. Munjie referred to one of them that was maybe a little bit different. I wanted to see the work I did get out to the world, and I was so naive. I thought if I published papers, people would just use them. It didn't work out that way. So I also wrote patents, and then I even got involved in starting companies, and as Sangeeta mentioned, licensing things to companies. And because of that, which actually had a lot of opposition and ridicule also, even at MIT back almost 40, 50 years ago, there are a lot of products based on this, all over the world, from companies we've started or worked with, ranging from new treatments for prostate cancer, endometriosis, schizophrenia, all kinds of things. Hundreds of millions of people use these. These are all like implants or microspheres, but if you didn't use them, the drugs would get destroyed. Even opioid addiction is up there from one of the things we started. But that being said, like I say, a lot of these might be microspheres or even implants like the drug-eluting stent, which I put up here. But the thing is that when you make particles smaller, you can, using the approaches we wrote in the Nature paper, it works, but sometimes they stick together and aggregate, and sometimes macrophages eat them. So working with Vladimir Torchelin and others, we had this idea you could take something in miralax, like polyethylene glycol, and coat these. And that was a paper we wrote 30 years ago, cited, I think, about 4,000 times. And you could even go further and put targeting molecules on these, which Omid Farrokhzad did in our lab. So I always explain this badly, but Nova, the TV show, they made a video of what we did, and it's so much better explanation than anything I could do. So here it is.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
Yeah, you can see why it does a better job than I do. I should say a lot of the clinicians I work with tell me it doesn't really blow the cell up like that. But the idea is you could use this for small molecules or for genetic therapy. You could use these approaches with lipids or polymers and so forth. And one of the things, of course, over the years, and we've worked on these for a long time, but if you look at various people, including Phil Sharp at MIT and others, they say, really, if you want to deliver genetic therapies, the biggest problem to solve would be delivery. And so over the years, I felt we could do that and even start companies in that. there were about 5 things that we did. One, showing that you could deliver nucleic acids back in that Nature paper, even though people said it was impossible. Then the idea of PEG. Then the idea of what's called ionizable substances, because if you make things charged, initially it might cause toxicity, but if you make them neutral at regular pH and then get charged inside the cell, they can get out of the endosome. Dan Pack, who's now at University of Kentucky, a postdoc of ours, and David Putnam showed this in the year 2000, and then other people advanced that in the lipid field as well. Then Dan Anderson, who was a postdoc with me, now full professor at MIT, had this idea of making thousands of different lipids, libraries, so you could have your choice of what ones are going to work well. And finally, Rohit Karnik, who was a postdoc with me, now a professor at MIT in mechanical engineering, had the idea of using microfluidics to make a lot of these. And so all these were published, something like 20 years ago. So I certainly, and we had patents on some of them, so I thought, well, you could create these products. So a couple companies, I thought I'd just give an example. This is from Alnylam, which Phil Sharp was a founder of, and I was on the founding scientific advisory board. This product was the first nanoparticle approved by the FDA for delivering RNA. It was actually done by Akinna Kink, who was an MIT student working in my lab, and now he's CEO of his own company. And then, of course, the other one that I should show you, which was alluded to by Sangeeta early on, is I helped start this company with Nubar Fayan, whose thesis committee I was on, and now on the MIT Corporation, and 2 others, to create nanoparticles that could protect messenger RNA. And of course, that led to the COVID vaccine, which is now used all over the world. And you see almost every other attempt at trying to make this vaccine didn't really work. Only 2 companies, Moderna and BioNTech, both using lipid nanoparticles and messenger RNA, got these approved. And that's the tip of the iceberg. You're going to see, I think, new cancer vaccines and other things. On the last slide, I just wanted to say areas that maybe we can do better on. And these are things that we're actually working on with funding from the Marble Center and from the Gates Foundation and with Bill Gates. And I'll just really list them. But reactogenicity, immunogenicity, could you make them less immunogenic so that if you gave a drug, you wouldn't get an immune reaction? More immunogenic, maybe having what's called adjuvant properties so that you could get even a better immune response. Longer shelf life, which Sangeeta actually mentioned in her very introductory remarks, so you can ship these all over the world. Anna Jakalenic in our lab, working with the Gates Foundation, has been doing a lot of work on that. Targeting to specific cells, which Dan Anderson and I have been working on. Other materials, there's all kinds of materials that may be useful. Right now, when you use lipid nanoparticles, people use 4, but maybe you don't necessarily need that many. And could you come up with things that might work better than PEG? Finally, could you cross barriers that right now you cannot cross, like the blood-brain barrier, which could be helpful in Alzheimer's and other diseases. So those are just a few of the things that we're thinking about and working on. And thank you very much for having me.
Thank you, Bob.
Well, thank
Amazing.
you.
You're going to maybe you can go all the way down.
Okay.
And then in the middle. Thank you. All right, well, for those of you not in medicine, hopefully that was a little bit of like rapid fire medical school education, and you're excited about what nanotechnology can do in medicine. This is a time for some Q&A from the audience. We have a whole bunch of things coming in on this fancy new app. I don't know what we call this. Let's see. Maybe we can start with, there's some pretty specific questions and interest on near-infrared wavelengths. Maybe for Muji and Angie, thinking about, what's the promise of near-infrared imaging? How far can things go? How could we make it better? Do we need new materials? What are your thoughts on that?
I think it's a very exciting field. I think there's a lot of opportunity because I think it's in early development. I don't know if you mean how far can it go in terms of how deep can it go. The deepest we've gone is about 8 centimeters, and it depends on the material type right now. It depends on the type of tissue. And do we need new materials? I think that we definitely need new materials. And I think that Munji showed some really exciting new materials around that area. We hope to be able to use it for actually ovarian cancer, breast cancer, and colon cancer in the next few years. And we're working on technologies that you'd be able to see it inside patients.
Great. Munji?
Yeah, so I think it's a very exciting area. I think the label-free work that you've done, I think there's a lot to do there as well. Just because, normal tissue doesn't really fluoresce in the near-infrared in the SWIR. But pathological tissues tend to have some signature there. So you can do multispectral imaging there and really see things. New materials would be great. Different kinds of optical tricks, like what you've done with the polarization, for instance. I don't know how deep you can go, but there's a lot of things you can do without going too deep. There are many parts of the body where you don't have to go more than a few centimeters in to really see things. So I think there's a lot to do there for sure.
Yeah. I think that's a great point. I mean, one thing that may not be on everyone's radar is the advances in surgery. You can actually get into most compartments now with robotic surgery or endoscopy, and so you could probably get pretty close to almost everything, actually, even for optics.
Yeah.
So there's another question. I think we talked about the excitement of getting these technologies into patients, and a challenge in medicine, which is appropriate, is that we have regulatory bodies that we have to collaborate with. And I'm curious, maybe starting with Bob first, how do you have advice for how to do that, or how has it changed? I know when we started the Cancer Center for Nanotechnology Excellence that you and Ralph co-led almost, I want to say 20 years ago.
Right.
Now, the NIH was trying to reform its approach to nanotechnology, and they started a toxicology service. How have you seen the regulatory bodies change their perspective or not over the years?
Yeah, well, I think that, it's a great question. I don't know that they've changed it that much. In an emergent, there's a couple times that, in general, they're very conservative. The idea is first do no harm. And so, and I think always the concern that you might do something bad is there. But there have been a couple of situations, both in the AIDS crisis back in the 90s and more recently with the COVID issue, where they approve things much more rapidly. I think at the end of the day, people make a decision, right, which is the risk-reward ratio, that when the risk is so high and so many people are dying and there's no real treatment, then they do things faster. In terms of advice, I think that what you do is you want to bring them in early rather than later. If you have a product that's different, it's good to talk to some of the people there or who used to work there. It's a long road.
Yeah. I think precedent and collaboration are really important. And as you point out, I think now that these materials have been in so many millions of people, at least that's a precedented material, right, that we can leverage for other applications. So you talked about cancer vaccines or other applications of those same 4 lipids. Hopefully the hurdles for them will be less high because they've been shown to be safe, at least in some applications. Muji, we were talking before the session about
the long road.
The long road, and also sort of parsing the different sections of the FDA, right? There's a drug division and a device division.
Right.
Can you tell us a little bit about how you...
For instance, the example of Lumicell was a drug-device combination where we invented a handheld device to be able to look within the surgical cavity for fluorescence that was generated by this agent, which was regulated under the drug division because it was an injectable and it was a relatively small molecule. It's a nanometer size, but still drug-like. And so those two regulatory bodies just don't talk to each other. And so the device division was extremely enthusiastic and tried to bring the drug division along, but they just, so it made it twice as hard basically, to get approval. But I think there is approval now, and there's a protocol that they've developed to talk to each other better, and hopefully in the future that will grease the wheels, the regulatory wheels.
Yeah,
It's a proof of concept that it can be done.
it will be a precedent. Angie?
I think that that in the case of Cision Vision, what Jeremy, the co-founder, did was quite quite brilliant, which is he developed a technology that in the end test still goes through regular pathology. So the technology that he developed was a way of getting samples more quickly into the pathologist's hands. So there wasn't the same kinds of FDA regulations. So the next step is for that technology is working on in humans and during surgery, but I don't think things will go as, invention-wise, I think they'll go as fast, but regulatory and acceptance will not be as fast as it was in that case. And he thought about this long and hard, and he was very smart in that.
Yeah, I think another opportunity in our space for early innovators is thinking about that clinical workflow and how to design your materials and really where in that process to be. You mentioned, you have to inject in the morning of the surgery, and you're talking about accelerating decision-making for pathologists, but not taking the pathologist out of the loop, which is super important. So I think that is a real important part of the ideation. We have a couple of minutes left, so maybe we could just do rapid fire. I'm going to ask you all what you're most excited about in nanomedicine. And I'm going to start with Bob and come this way.
Well, I mentioned one of them in the talk, like target, can you target, right now you can target nanoparticles to the liver pretty easily, but not to really other cells or other parts of the body, including cancer cells. So, I'd be excited about that. So Dan Anderson and I are working on those kinds of things.
Great. Angie?
I love working directly with the surgeons, and I love working with patients. I see them in clinic. I see them before and after surgery. I love getting to know them. And it makes me even more passionate about really trying to make an impact on ovarian cancer, and that is what I'm most excited about, is trying to impact patients' lives in a positive way. As an engineer, I never thought that would be possible. But you're a physician and engineer, so it's possible, but so I'm excited about that.
Amazing.
So I would say that working very closely with the clinicians, you actually are both, so you can work closely with yourself. Learning what the specifications of what you're trying to do have, those are super important. You can invent the greatest thing, but like this injectable, if it takes 3 days, it's a non-starter.
Right.
That we did. And so knowing ahead of time, what are the constraints so that you can design around the constraints, I think, and we know how to do that in the nano world. We just have to make sure that we talk to the clinicians ahead of time before we start inventing things.
Yeah. Well, I think all three of you are great examples of team science and why we're on such an important precipice for the field. So thank you very much.
Thank you.
Thank you, Sangeeta.
Thank you, Sangeeta.